Good morning. In the last class, we discussed about the adaptations in C4 plants to avoid photorespiration. As you know that photorespiration is a highly wasteful process because it releases the carbon dioxide which have been fixed by C3 cycle, and it also uses energy. So this process must be avoided, and in order to avoid this process, there are some adaptations in the plants which are living in hot conditions, high temperature conditions. And these adaptations we have already discussed, that is presence of dimorphic chloroplast and the presence of bundle sheath cells or the Kranz anatomy. And we have seen that in case of C4 plants, two types of chloroplast are there, granum and agranum. Granum is with thylakoids, agranum is without thylakoids. The granum chloroplast is present in the mesophyll cells where light reaction will take place and oxygen will be released, whereas agranum chloroplast is located in the bundle sheath cells, and in the bundle sheath cells no light reaction will take place and no oxygen release will be there. And in case of C4 plants, the site of C3 cycle has also been shifted from mesophyll cell to the bundle sheath cells. Rubisco is shifted from mesophyll cell to the bundle sheath cell. So C3 cycle will take place in the bundle sheath cell. In case of C3 plants, the C3 cycle or the Calvin cycle takes place in mesophyll cells, while in case of C4 plants, C3 cycle will take place in bundle sheath cells because the site of Rubisco has been shifted from mesophyll cells to the bundle sheath cells. Now today we will discuss C4 cycle. So let's start with the C4 cycle. The cycle is called C4 because the first stable product which you get in this cycle is a 4-carbon compound and that is your oxaloacetic acid. Oxaloacetic acid. So that's why the cycle is called C4 cycle, and it is also called as Hatch-Slack pathway. It is named after the scientist, Hatch-Slack pathway. Now what happens in C4 cycle? In C4 plants we have two types of cells, right? We have mesophyll cells and bundle sheath cells. जो आपके C3 plants होते हैं, they are the plants of cool and wet areas, while C4 plants are the plants found in hotter regions. So here we have mesophyll cells and then we have bundle sheath cell. So C4 cycle will be completed in these two cells. And the main aim of this C4 cycle is to concentrate the carbon dioxide in bundle sheath cell. Because in bundle sheath cell Rubisco is present and we want to start C3 cycle with that Rubisco. And for C3 cycles to start, we need carbon dioxide, right? So the main aim of C4 cycle or the Hatch-Slack pathway is to concentrate the carbon dioxide in the bundle sheath cell so that carbon dioxide can be accepted by RUBP and it can initiate the C3 cycle or Calvin cycle for the formation of glucose. C3 cycle is universal. It will take place in all the plants, whether they are plants of hot region or the cool regions, but C3 cycle is universal because this is the only method to produce glucose in the plants. Okay, so C3 will take place in all the plants. Now how C4 cycle starts? Carbon dioxide will enter inside the mesophyll cells through the stomatal pores. Mesophyll cells are present in the leaves. Okay, bundle sheath cells are also present in the leaves. So carbon dioxide is going to enter inside the mesophyll cell through the stomatal pore, and inside the mesophyll cell it will combine with water to form carbonic acid, that is H2CO3. This H2CO3 or carbonic acid, okay, this is carbonic acid. This H2CO3 can dissociate to give bicarbonate ions and hydrogen ions. Now this bicarbonate ion, that is HCO3 negative, can combine with 3 carbon compound that is called PEP. Okay, PEP is a 3 carbon compound. It contains 3 carbons. Its name is phosphoenol pyruvate. Bicarbonate ion contains 1 carbon. Phosphoenol pyruvate contains 3 carbon. So these two together will form 4 carbon compound that is called oxaloacetic acid. Okay, it contains 4 carbons. As this is the first stable product of the cycle, so the cycle is called C4 cycle. Now this oxaloacetic acid is converted into malic acid, which is again a 4 carbon compound. Oxaloacetic acid cannot enter inside the bundle sheath cells because the membrane is impermeable. So oxaloacetic acid is first converted into malic acid. This malic acid will now enter inside the bundle sheath cells. Malic acid is also 4 carbon compound. So now we have malic acid in the bundle sheath cell. This malic acid is now decarboxylated. Decarboxylated means the carbon dioxide will be removed from the malic acid. So here carbon dioxide is removed. Malic acid contains 4 carbons. If 1 carbon dioxide is removed, 3 carbons are left behind, and those 3 carbons will help in the formation of pyruvate or pyruvic acid. Okay, it is a 3 carbon compound because 1 carbon has been released as carbon dioxide and 3 are left behind. So 3 carbons will result in the formation of pyruvate. This pyruvate is converted into PEP, that is phosphoenol pyruvate, and the PEP will go back to mesophyll cell so that new cycle can be initiated, because we need to produce more carbon dioxide, right? So we need new cycles for that. So PEP must be regenerated. So this pyruvate is converted into PEP, and PEP will go back to the mesophyll cell to accept new carbon molecule and to initiate the cycle. So this is your C4 cycle. Now what about this carbon dioxide which is released here? This carbon dioxide is accepted by RUBP, which is a 5 carbon compound, ribulose 1,5-bisphosphate, and they will enter in the C3 cycle. Okay, so C3 cycle will begin here. So in case of C4 plants, C3 cycle is taking place in the bundle sheath cell. Now if we compare the productivity of C3 and C4 plants, then C4 plants are more productive than C3 plants, because in C3 plants there is photorespiration, which is a highly wasteful process, right? So it is wasting the carbon dioxide which has been fixed by the C3 cycle. But in case of C4 plants, there is no photorespiration. Why there is no photorespiration? Because they have certain modifications and adaptations in their body, like they have two types of chloroplast and bundle sheath cells, and the site of Rubisco or C3 cycle has also been shifted from mesophyll cell to the bundle sheath cell. So there is no photorespiration. The oxygen will be released in the mesophyll cell only, because the light reaction is taking place in the mesophyll cell only. And whatever oxygen is produced here in the mesophyll cell from the light reaction will not be able to enter inside the bundle sheath cell, because bundle sheath cells are impervious to the gaseous exchange. Okay, so oxygen will not be able to enter inside the bundle sheath cell, thus avoiding the photorespiration. Bundle sheath cells contain A granum chloroplast, so no light reaction will take place here. No light reaction means no oxygen, no oxygen means no photorespiration. And we have to increase the carbon dioxide concentration here so that C3 cycle can take place here. And this carbon dioxide concentration is increased by C4 cycle. The carbon dioxide which is entering from the atmosphere is fixed with the help of phosphoenol pyruvate, thus forming 4-carbon compound oxaloacetic acid. This is then converted into malic acid. Malic acid enters inside the bundle sheath cells, where it is decarboxylated to release carbon dioxide and forms pyruvate. Pyruvate is converted into PEP again, and it is sent back to the mesophyll cell, whereas this carbon dioxide enters in the C3 cycle to form glucose. So this is C4 cycle. In the next video, we will discuss about cressolation acid metabolism or CAM plants.